ATP, water, and carbon dioxide.
This process is an exergonic reaction, meaning that it releases energy through anabolic and catabolic pathways. The overall equation for cellular respiration is C6H12O6 plus 6O2, which yields 6CO2, 6H2O, and ATP.
The process of cellular respiration is broken down into 3 steps: glycolysis, the Krebs cycle, also called the citric acid cycle, and the electron transport chain. This is the overall equation for cellular respiration. Glucose and oxygen are the reactants, which means they're consumed or used during the reaction. So glucose is used during glycolysis, where it's broken down into 2 pyruvates. The oxygen is used during the electron transport chain, as it is very electronegative and pulls the electrons down the chain. Over here, we have our 2 reactants. On this side of the reaction, we have the products: carbon dioxide, water, and ATP. There are about 38 molecules of ATP produced, 6 molecules of carbon dioxide, and 6 of water. The carbon dioxide is produced during 2 different parts of cellular respiration. First, during the pyruvate oxidation, and secondly, during the citric acid cycle. Water is produced during the electron transport chain as the hydrogens of the mitochondrial matrix attach to the oxygen that is sitting at the 4th protein complex. ATP is produced during glycolysis, the citric acid cycle, and during chemiosmosis.
Glycolysis is an anabolic step to cellular respiration, which literally means sugar splitting. In this process, one 6-carbon sugar, known as glucose, gets broken down into 2 3-carbon sugars, known as pyruvate. This process can be done aerobically or anaerobically and has 2 phases to it: an energy investment phase and an energy payoff phase. I will further explain these phases in the next videos. The first part of glycolysis is the energy investment phase. Step 1, glucose enters the cell and is phosphorylated by the enzyme hexokinase. Glucose 6-phosphate is then transformed into fructose 6-phosphate. These 2 are isomers of one another. This is done through the enzyme phosphoglucoisomerase. Step 3 of the energy investment phase, phosphofructose kinase transfers a phosphate group from ATP to sugar. A second ATP is invested into glycolysis. Sugar is now ready to be split because 2 phosphate groups total are present. In step 4 of the energy investment phase, aldolase splits glucose into 2 3-carbon sugars known as pyruvate. Dihydroxyacetone phosphate and glyciderol phosphate form, and they are isomers of each other. Lastly, in step 5, isomerase catalyzes reversible reactions between the 2 pyruvates. An equilibrium is never reached because glyciderol aldehyde 3-phosphate is only used as its substrate. Step 6 of glycolysis begins the energy payoff phase. An important enzyme known as triose phosphate dehydrogenase catalyzes 2 sequential reactions while it holds glyciderol 3-phosphate in its active site. Glycideraldehyde 3-phosphate is oxidized due to a transfer of electrons in H+ to NAD+, forming NADH, which has a lot of potential energy. Glycideraldehyde 3-phosphate is now 3-biphosphoglycerate. In step 7, for each glucose molecule, 2 ATP molecules are formed from 2 ADP and 2P. ATP has been repaired. 3-biphosphoglycerate is now 3-phosphoglycerate. The enzyme that allows step 7 to occur is phosphoglyciderokinase. Phosphoglycideromutase relocates a phosphate group, which prepares substrates for the next reaction. The enzyme that allows step 9 to occur is enolase. Enolase yields phosphoenolpyruvate, which is known as PEP, by the hydrolysis of a substrate. This results in high potential energy, allowing for the next step to occur. The enzyme that allows the last step of glycolysis to occur is pyruvate kinase. A phosphate group goes from PEP to ADP to produce ATP. This happens twice.
After glycolysis happens, there is a conversion that must occur in order to connect glycolysis to the citric acid cycle. This conversion is of a single pyruvate into acetyl coenzyme A. This conversion must take place 2 times because there are 2 pyruvates produced for every 1 glucose. Since the pyruvate is a charged molecule, in eukaryotic cells, active transport happens via a protein. The pyruvate dehydrogenase complex catalyzes 3 steps: the formation of CO2, the reduction of NAD+ into NADH plus H+, and the addition of coenzyme A. Only after these 3 steps occur will acetyl coenzyme A be made and thus enter the citric acid cycle. Since the pyruvate was fixed into acetyl coenzyme A, now we're in the mitochondria and the citric acid cycle can begin. Here is a diagram of a simplified version of the citric acid cycle. I will next further explain each of the steps. Acetyl coenzyme A adds its 2 carbon acetyl groups to oxaloacetate, producing citrate. Citrate is converted into its isomer, isocitrate, by the removal of 1 water molecule and an addition of another. Isocitrate is oxidized, reducing NAD+ to NADH. Also, a carbon dioxide molecule is lost. Another carbon dioxide molecule is lost, and that resulting compound is oxidized, reducing NAD+, again, to NADH. Also, the remaining molecule is attached to coenzyme A by an unstable bond. In this step, GDP is turned into GTP by the addition of a phosphate group. This phosphate group kicks out coenzyme A. Sometimes, because this additional phosphate group, ADP, turns into ATP. GTP and ATP have similar functions. 2 hydrogens are added to FAD, thus producing FADH2. Also, it oxidizes the succinate. In this 7th step, the addition of a water molecule rearranges the bonds of the substrate. The substrate is then oxidized, reducing NAD+ into NADH and regenerating oxaloacetate. After the oxaloacetate is regenerated, then the cycle can begin again. As Grace explained earlier, glycolysis breaks down a single glucose molecule into 2 pyruvates. The citric acid cycle that I just explained was for a single pyruvate. So for 1 glucose molecule, the citric acid actually can produce 2 molecules of ATP, 2 molecules of FADH2, and 6 molecules of NADH. This is a diagram of the mitochondria. The mitochondria is home to 2 of the 3 steps of cellular respiration. The citric acid cycle and the oxidative phosphorylation steps both happen in the mitochondria. Here's the outer membrane in purple. The inner membrane is in the green. The inner membrane space is in the red. The matrix is in pink. And the cristae are these little folds in the inner membrane. For the citric acid cycle, it takes place in the matrix of the mitochondria. For the oxidative phosphorylation, for the ETC step of that process, it takes place in the green inner membrane part of the mitochondria. Fermentation and anaerobic respiration enable cells to produce ATP without the use of oxygen. The difference between fermentation and anaerobic respiration is that fermentation, there is no electron transport chain used, and in anaerobic respiration, the electron transport chain is used. For fermentation, there's two types. The first is alcohol fermentation, and the second is lactic acid fermentation. I will explain the two of those later. When comparing fermentation and aerobic respiration, there are some similarities. Both produce ATP by harvesting chemical energy from food. Both use glycolysis to oxidize glucose and other organic matter with a net production of 2 ATP. And both use NAD+ as the oxidizing agent that accepts the electrons. There is one major key difference that I will explain. In order to understand the difference between fermentation and aerobic respiration, you need to understand and answer the question, how is NADH oxidized back to NAD+? So you have to look at what is the final electron acceptor. For fermentation, if you're doing alcohol fermentation, the final electron acceptor is ethanol. In fermentation, if you're doing lactic acid fermentation, that final electron acceptor is acetaldehyde. But when looking at aerobic respiration, the final electron acceptor is oxygen. Because oxygen is present, the process not only regenerates the NAD+, but it also pays an ATP bonus when the stepwise electron transport from the NADH is oxidized, drives the oxidative phosphorylation. In aerobic respiration, unlike in fermentation, the electron transport chain is present, and it has oxygen as its final electron acceptor. Because oxygen is very electronegative, the electrons are pulled very, very strongly down the electron transport chain, making the aerobic respiration process way more efficient than the fermentation or anaerobic processes. Aerobic respiration is able to produce about 19 times more ATP than fermentation or anaerobic respiration. Here's a diagram of alcohol fermentation. It starts with glucose going through glycolysis with a net production of 2 ATP, 2 NADH, and 2 pyruvates. This is when fermentation begins. So for alcohol fermentation, 2 carbon dioxides are released from the 2 pyruvates, resulting in 2 acetaldehydes. Then the acetaldehyde is reduced by 2 NADH, which thus produces 2 ethanols. Here's a diagram of lactic acid fermentation. It starts with glucose going through glycolysis, producing a net of 2 ATP and 2 NADH. Here, the 2 pyruvate are directly reduced by the NADH to produce 2 lactate. Oxidative phosphorylation is the metabolic pathway in which cells use enzymes to oxidize nutrients, thereby releasing energy in which is used to produce ATP. This is the part of the cellular respiration in which the most ATP is produced. About 34 molecules of ATP are produced. Oxidative phosphorylation consists of 2 parts to oxidative phosphorylation. The first part is the electron transport chain, and the second part is chemiosmosis. This diagram shows the electron transport chain, which is the first part of the oxidative phosphorylation step. NADH and FADH2 shuttle high-energy electrons extracted from food during glycolysis and the citric acid cycle to the electron transport chain that is built in the inner mitochondrial membrane. The blue arrows trace the transport of electrons. Just stop. There are 4 protein complexes of electron carriers in the electron transport chain. 1, 2, 3, and 4. There are also 2 mobile carriers, ubiquinone, which is Q, and cytochrome C, which is CYTC here. Q and CYTC shuttle the electrons back and forth from 1, 2, 3, and 4. As complexes 1, 3, and 4 accept and then donate electrons, they pump protons from the mitochondrial matrix into the inner membrane space. In prokaryotes, protons are pumped outside the plasma membrane. FADH2 deposits its electrons via complex 2. It results in fewer proteins being pumped into the inner membrane space than that occurs for NADH. Chemical energy originally harvested from food is transformed into a proton motive force, a gradient of H+ across the membrane. That finishes the electron transport chain. Now we can move on to the second part of oxidative phosphorylation, chemiosmosis. Here's a diagram of chemiosmosis, the second part of oxidative phosphorylation. During chemiosmosis, the protons flow back down their gradient via ATP synthase, an enzyme which is built into the membrane nearby. The ATP synthase harnesses the proton motive force to phosphorylate ADP, forming ATP. Together, the electron transport chain previously explained and chemiosmosis make up oxidative phosphorylation, and together produce the largest amount of ATP of cellular respiration. For every 1 glucose molecule of cellular respiration, about 36 to 38 molecules of ATP are produced. ATP stands for adenosine triphosphate. So this is a diagram of ATP. So there's a nitrogenous base called adenosine. There's a sugar called ribose. And there are 3 phosphate groups, each right here. In between each bond of the phosphates, there's energy. Here, there's a little energy. Here, there's a moderate amount. And in between the last 2 phosphates, there's a lot of energy. So when ATP loses a phosphate group, it turns into ADP. And as I said, since there's a lot of energy in between the 2 phosphate groups, when this one is broken off, it releases a lot of energy. So then you have an ADP molecule, which stands for adenosine diphosphate, because there's only 2 phosphate groups. In order for ADP to turn into ATP again, to get a lot of that energy again, you have to add on a phosphate group. And then it is able to turn into ATP again. ATP drives metabolic processes, which is why it is so essential to life.
Thanks for watching!